So now, having learned about the parameter inference problem and the advantages of Bayesian inference for biological models, we will jump into a Python toolbox called BioScript that implements parameter inference for biological systems. So you can import biological models in an SPML format. Those can be created in any tool of your choice. And you can specify then what parameters you're trying to estimate. You can specify the experimental data that you have. Once you have done all of that, Bioscrape calls another package called Python Emcee. That's the package for Markov chain Monte Carlo sampler. And you don't really need to know anything about that package. All you need to know is load your data, load your parameter values, priors, and set it up. So Bioscript runs the simulations internally and then gives you out the parameter distributions as well. So we will learn how to use it with few notebooks, Jupyter notebooks, that will go through different examples that are commonly encountered when you're running parameter inference. So if you want to run along with these notebooks, you can download the notebooks from the link I posted. And you need to also install Bioscrape. So you can get it from GitHub. You need to update to the latest version by doing git pull origin master. So this pulls the latest code. And then you can install the software from the Bioscript main directory by running python setup.py install. I'm assuming that you have the software installed. And if you're trying to run, it's fine if you're not trying to run the notebooks. You can look at my screen as I go through all of these notebooks. So the first example that we will look at is an example for linear model, where we have linearly, we want to fit this data that we are seeing using just this equation, y equals to mx plus b. So it's a very simple, basic one to get you started with how everything is set up. It's not even a dynamical model because it's just a simple equation. Okay, so for the linear model, I jumped into this Jupyter notebook titled Linear Model that I linked earlier. So let's go through this. The first cell is just the plotting setup. The second one is what we can start to look at closely is, so second one, I'm creating that linear model. And since I'm doing it with Bioscript, you can actually skip it for now because this is not what you will be usually doing. What you will have is biological model ready with you in an SPML format to just import it. And that example will be clear in other notebooks. But here, I'm creating that linear model within Bioscript itself. That's another capability of Bioscript. So I'm creating this y equals to mx plus b kind of model in this model object and running the simulation for it. When we have that model, what we can do is we can generate the artificial data that you saw around that model and then fit that artificial data to that same model to see if everything is working. So I generate this artificial data. So these dots here are all the data points, and the gray line is the model. So every experimental data in Bioscript inference has to be loaded in as pandas data frame. And so I create a pd.data frame, that's the pandas data frame, and then load in the data into this data frame. So my x, I load in as x0, y is the experimental data y that I created earlier. So a pandas data frame then looks like this. You have two columns, x and y, for the model y equals to mx plus b. So scroll down, and now that's it. Now we can directly use Bioscript inference. So to use Bioscript inference, all you need to do is call bioscript.inference and then import PyInference function that we will use for parameter inference. And it can take in all of the different parameters that you need to choose. So the first thing here is prior, and this is what we just talked about, is that you need to tell the sampler or the algorithm, you need to help it out by saying what your parameters might look like. So this is the information that you already have. Of course, you can just say that I don't know anything about my parameters and just run it. That is also fine, but then you are brute forcing it and you're forcing the algorithm to explore all of the space. For simpler models, that will work just as fine. You won't even observe a difference. But when you go into more complicated models, you will need to choose priors intelligently to push the algorithm to the right places, so to speak. So here, what I'm doing is I'm giving Gaussian priors for both parameters M and B. The syntax of how it works is prior is a dictionary with key entries where key entries are all the model parameters. So here M and B are the keys, M and B. And for each key is associated a list, which specifies what kind of prior it is, and then specifies the parameters that are needed for that prior. So for M, the first entry always needs to specify what kind of prior it is. So here we see that we are trying to say that this is a Gaussian prior. And then this second and third parameter are according to the Gaussian prior. Because I chose the Gaussian prior, I need to specify the mean and the standard deviation for the Gaussian distribution for this parameter. So with this whole thing, what I'm saying is that I know that the M parameter might lie in this Gaussian distribution of mean M true, the true value of the slope, and with the standard deviation of 500. So I'm telling the sampler algorithm to explore according to this space, according to this distribution, that these values are more likely, but there is a 500 standard deviation. Similarly for B, we have a Gaussian distribution that we want to specify, and the mean and the standard deviation. So this completes the prior description, the prior probability distribution for the model. Note that we are not writing down the probability distribution for the priors exactly, and that is why you don't see the log prior that was hidden when we saw in the lecture, that what we actually do is we do log prior plus log likelihood. All of that is done internally for you. You don't need to worry about that. All you need to do is specify what kind of probability distribution your parameters might lie in. If you don't know anything about those parameters, you can just say that those are uniform probability density. So you're just saying, pick randomly from this maximum and minimum range of values, and that's fine. For a simpler model, that works great as well. The next line is the main line and the only line that does everything, but takes into account all of your things and sets the correct objects to run the parameter inference. And that's called the pi inference function. The first argument for that is the model. So this model M is the Bioscrape model that you can load in or you can create. So here we created, but we will see in the next examples that you can load SPML model as well. So you load in that model, specify it here. The second argument is EXP data, is experimental data. And this is a data frame that we created, this one that we see here. The third one is measurements. So it's all pretty self-explanatory now. It's what are the list of species that you're measuring. So that specifies the number of outputs. Of course, this can be multiple as well. But then you have to shape your data accordingly if you have multiple outputs. The time column specifies what the, and I call this the time column because in biological models, you usually have time course data. So this is essentially, this is just the x-axis. So here, this is the x-axis and this is the y-axis. Then you specify the params to estimate, so the parameters that you want to estimate. And this is again a list of names of parameters that you want to estimate. And now here are the parameters that we talked about in the lecture for the MCMC sampler. That is, n walkers is how many walkers you want. That is how many different walkers you want to start independently with some number of steps that you want to look at for convergence at the end. And second is steps, is how many steps you want each walker to run. The third is init seed, is something that you can also ignore, it's optional, but it's just how big of a ball around your initial guess do you want to start with. So you give some initial values that are set in the model directly, and the sampling algorithm starts with those initial values that you have set. And sometimes the sampling algorithm is very sensitive to where you start, because if you might be starting from a region where it's very hard to go into or converge to this valley of the correct or optimized posterior probability density, it might take a very long time, or it might not even be possible. So it's important that you set the initial values correctly. And then the initial seed is how big of a ball you start with from the initial values to start the sampling algorithm, but it's optional, so you can ignore it as well. The next is prior. So you specify what the prior is. We already created the dictionary, and I explained how the dictionary should look like. So that goes in here. The next is sim type. So what kind of simulations do you want to run underneath? That's deterministic or stochastic. And then others are, again, optional arguments that you can skip. So plot show is if you want to show the plots or not, and convergence check if you want to check for convergence or not. Once that's done, you run it. So it runs and shows this progress bar with number of steps, and then plots the data, because I have plotted it here, although I did false here, but I used the returned output object, PID, and called the plot MCMC results function to plot the sampler output, which is stored inside sampler. So sampler stores all the samples, and this is an Emcee object, Python Emcee package object. PID is a Bioscript object that contains all of this information that you just passed in. And so I plot the MCMC results, and this is what I have here. Now we can look at this closely. So you see, we started with some value of 0.7 here, and we were looking at exactly this progression of the MCMC algorithm for this parameter earlier, but we were only looking at a very few steps, maybe. So we were not seeing this exploration, right? So if you just stopped looking here, you would get the wrong answer. So if you just stopped looking at, say, 20 steps or 50 steps, or maybe even like 100 steps, right? So if you stop at 100 steps, what you see is it doesn't really explore this space on top. But if you let it go on for longer than that, you see that actually, although it started there, explored this space, then it actually found an even minimal cost function in this space. And so then all of the walkers are exploring this space now. And then some go wild and go out, but then they come back because they find that the cost values are very high over there. So that's what's happening here. So for each walker is doing this independently. So we have 32 walkers here, and so all of those 32 are doing this independently. And what we see is that looking at this, we can say that we have sort of convergence, that if we draw some samples now out from this randomly, we will get a representative sample for the posterior probability density for M. Similarly, we have the other parameter B. It started with some value 4.2, but it actually converged down to some other value that you see here, and then we have this chain for B as well. So the number of parameters you estimate, you get these chains for every parameter. And then another useful plot that comes out of this is this one, the corner plot, which gives the actual distributions plotted out. So here on the x-axis is M and B. So this is the distribution for how M looks like. The values are here. And this is the distribution for how B looks like. And on the corner, we have the correlation of both parameters together. That can be used to see where your overall posteriors for both together are skewed. So here, you can use those to choose the final values, for example, that you want to use in your model or something like that. completes the parameter inference for this model. And now we can do various kinds of plotting. So one is, as I said, you want to draw a representative sample from this, right? That represents the probability distribution. And you can do that easily because you have access to all of those samples in this sampler object. So I do that exactly here and plot the results. So what I do is from the sampler, I get the objects. I discard the first 100 steps. So as you can see, first 100 steps are what's called the burn-in period. So that's the time that it's taking to settle down to the actual values. So those are garbage for us because that was just the part where it was exploring the space. So we don't really need to take it into our account. So this is, again, this is Python Emcee function. So I use the sampler to get chain, discard the first 100. 10 is how many samples you want. Like, so 15 means at every 15 samples, we'll just take that sample from here. We can read up more again for Python Emcee and then give a flat chain. So this is flat samples for the parameter values. So this just gives you a list of parameter values. So this is how you draw that representative sample that I have been mentioning again and again. So you have the sampler, you discard the first few, and then you sample out some values that you get out in this flat samples that you can then use to set to the model and then simulate. That's what exactly I do. And I can run this plot to see that what the MCMC is giving us is, instead of this line here, it's the slope and the intercept both have changed a little bit. And these orange plots are all the different possible plots from the representative sample. And remember that this is Bayesian inference, so you're not finding out one exact value for a parameter. What you're doing is finding out a distribution of values that are possible. And that is why what you're getting out is a distribution of parameter model simulations that are possible. You can read up more utility functions that are available, and that's it. We can quickly go through the other examples. I know we are late on time, but that's fine. We can go through the other examples quickly now. Yeah, so we were at linear model. We did this already. Now let's look at a birth-death process. It's just, again, a very simple model, but we are going up, we're increasing up the complexity. This one is one species model, but it's a dynamical model now. It gets produced and gets degraded. We have the model and we create the artificial data by adding some noise to the model, similar to how we did the other one. And then we want to estimate the parameters if the data looked like this in the red curves. So, birth-death model. We can ignore the first few, it's exactly the same. Here, where everything is important. So we import the SPML model. So that's the toy SPML model.xml. That's the file that has my model. I can create this model from any place. And then I import it to Bioscrape. The second line again is important, is now I'm reading the data from a CSV file. So I have written that data to a CSV file already, and I'm reading it using pandas, pd.readcsv, and to create this data frame. Once that's done, I create the prior object for one parameter, say if I only want to estimate parameter D1, and I specify that I want a Gaussian prior. All the other setup is more or less the same. And if I run this, what I see is something like this. So I had the initial setup at 0.275. It explored, kept exploring, and then if you see all the walkers, I only had 5 of those, but to make it run quickly. And it's actually enough because we see that all 5 converge to this very small space around 0.175. And we get the probability distribution for that parameter. This was just one parameter in the model because there is the birth rate and the death rate. So this is just for the death rate. The model, to remind you, looks like this. The birth rate is K1 and the death rate is D1. We can identify the distributions for both those parameters. To do that is simple as well. Just specify the priors for both now and specify that you want to estimate both and then run it. What you see again are the same progression of those parameters. And then the distribution is now a little bit different because it's skewed towards the initial values. Okay. And similar to the linear model example, you can plot the results by sampling from that sampler chain of values, and then that's it. Moving more useful, third example is when you have multiple initial conditions, for example, for species and inducer concentration that might be varying. So you have this data where you're varying some initial concentration of a species, and you observe this data for you have 5 different trajectories. So how would you estimate the parameters for the model then? So the important thing to note here is that the likelihood computations now take into account all 5 together at every step. So for every new parameter set that it proposes, the sampler, it computes the likelihood for this first one by subtracting it from the data, and the simulation is this one, say, or second one, and the third one, and the fourth one, and all of those costs are added together. So it tries to fit all 5 together. Of course, you can do it individually and you can do it different ways, but you can also try, and this is more realistic as well, because you want your model not just to fit one initial condition, but you want the parameter values so that they fit most of the initial conditions, or as many as the initial conditions as you have here. So let's look at the setup for this. And the initial setup is just exactly the same, importing the model. The only part where it's different is, so this is generating the experimental data. So now the experimental data looks something like this. Oh, this is multiple trajectories for the same model. This is not multiple initial conditions, sorry. So yeah, I specify here number of trajectories, 10. And that's why instead of just having one data trajectory, I have 10 different trajectories that have noise in them. So these red are all the trajectories that I have generated by adding this noise here. And now I can pass in, so the data looks like this, where I have one axis for time points and 10 different trajectories for the same time points. And this is, again, a very common use case. You are measuring one output, but you are doing it 10 times. So you have 10 different trajectories for it. So your data frame should look something like this. And then you can create your experimental data, EXP data object, as a list of data frames, as I mentioned earlier. So it's a list, empty list. And in that list, what I'm putting is data frames. So I'm creating these data frames by reading the CSV file. And that's it. A list of data frames can be passed into BioScript inference. So that directly goes into the PyInference function here. All of the other setup is the same, and it runs the algorithm again. So now again, we are seeing this converge to certain values, and you have the distributions. The next example that we saw was with multiple trajectories when you have multiple initial conditions. So let's say we have the same model, but now you have certain initial condition I that changes this rate K1I, right? It's a hill function here. So you could think of this as inducer affecting the birth rate of X. So for this model, again, the setup is exactly the same. You create this model, import the model, and this is where I'm generating the experimental data artificially. So I have 4 different trajectories for different initial conditions, and then adding noise to it here. So my CSV looks like this again, time points and 4 different trajectories. These could be 4 different initial conditions. And now the important thing is to create your list of initial conditions, and this needs to be passed in. So you need to tell the algorithm how the data is shaped. So this list, the first element should be the dictionary of initial condition that corresponds to the first in the experimental data. So X0 corresponds to the first initial condition. I is 10, that is the initial condition is 10, corresponds to X1 column here, and so on. And so exactly corresponding to the experimental data list of data frames. So you have data frames like this, right? First data frame, And the second one is x1 with time points, x2 with time points. So you have 4 data frames. So you have 4 different initial conditions. And you pass in experimental data, and now we also need to pass in the initial conditions in the same function. And this is a list. And you can run the algorithm again, and it tries to find out what are the parameter distributions that it converges to. So it started with some value 50, but it actually find out now that some value around 57 or 5960 is actually what's correct. And you can plot so you can see for all the different initial conditions, it tried to move up the simulation. So it changed those values. So it tried to get the data, but you were only estimating one parameter. What we can do is estimate all 3 parameters of the model. And so then we get 3 different chains, 3 different distributions. And you see that the overall fits improved when you try to fit all 3 values. With that, I would like to invite you to look at all of these notebooks yourself. Those are fairly documented. You can just read through it, should be clear. There are other notebooks like stochastic inference, how to do that, how to work with the internal objects. And you can just pick up this BioScript inference and give it a try with your own SPML model and some experimental data that you might have. If you don't, you can just generate some artificial data like I explained in the notebooks that I just showed, and try to run parameter inference on your own SPML model. That would be a very helpful exercise, as you will find out, because then that gives you a much better idea of where your parameters are and what your model performs like. Thank you so much for your attention.